One day, Carlos the carbon dioxide molecule was floating through the air. Suddenly, he was absorbed into Larry the leaf through his pores.
Hey man, what's going on?
You're going through a process called photosynthesis, which will help to give me energy. Photosynthesis starts with you and water.
That's cool, dude. I ain't even mad.
Carlos entered the leaf, where he then went into the cells. Inside the cell, he met Christina the chloroplast, who was notorious for absorbing CO2 molecules like himself.
I need to absorb you so that can get energy.
In the chloroplast, Carlos met Walter the water molecule.
Oh, hey Walter, how's it going?
I'm scared to be a part of photosynthesis.
It's okay. We can talk to Thor the thylakoid together.
They approached Thor the thylakoid and said,
Oh, hi. Hey, how are you doing?
Hi. Thor started telling Carlos about what he does.
Photosystem 2 absorbs light energy, allowing the P680 reaction center of two chlorophyll A molecules to donate an electron to the primary electron acceptor. The reaction center chlorophyll is oxidized and now requires an electron. An enzyme splits a water molecule into two hydrogen ions, two electrons, and an oxygen atom. The electrons are supplied to the P680 chlorophyll A molecules. The oxygen combines with another oxygen molecule, forming the O2 that will be released into the atmosphere. The original excited electron passes from the primary electron acceptor of photosystem 2 to photosystem 1 through an electron transport chain, similar to the electron chain in the cellular respiration. The energy from the transfer of electrons down the electron transport chain is used to pump protons, creating a gradient that is used in chemiosmosis to phosphorylate ADP to ATP. ATP will be used as energy in the formation of carbohydrates in the Calvin cycle. Meanwhile, light energy has also activated PS1, resulting in the donation of an electron to its primary electron acceptor. The electrons just donated by PS1 are replaced by the electrons from PS2. The primary electron acceptor of photosystem 1 passes the excited electrons along to another electron transport chain, which transmits them to NADP+, which is reduced to NADPH, the second of the two important light reaction products. The high-energy electrons of NADPH are now available for use in the Calvin cycle.
The other part of photosynthesis is the dark reaction, or Calvin cycle. Larry the leaf needs both the light reactions and the Calvin cycle in order for photosynthesis to occur.
Biphosphate. These reactions are catalyzed by the enzyme Rubisco and produce an unstable product that immediately splits into two 3-carbon compounds called 3-phosphoglycerate. At this point, carbon has been fixed, the incorporation of CO2 into an organic compound. The 3-phosphoglycerate molecules are phosphorylated to become 1,3-biphosphoglycerate. Next, 6-NADPH reduce the 6,1,3-biphosphoglycerates to 6-glyceraldehyde 3-phosphate. Once glyceraldehyde 3-phosphate leaves the cycle to be used by the plant cell, two G3P molecules can combine to form glucose, which is generally listed as the final product of photosynthesis. Finally, ribulose biphosphate is generated as the 5-glyceraldehyde 3-phosphates are reworked into 3 of the starting molecules, with the expenditure of 3 ATP molecules.
So hot in here. Hi, I'm Carlos, the carbon dioxide molecule.
I'm Daisy, the desert.
So, I just went through photosynthesis in a leaf, and now I'm ready to do photosynthesis in the desert, where it's hot and arid.
Good thing plants in the desert use C4 or CAM for photosynthesis.
Oh, that's cool. How does that work?
Well, C4 and CAM have metabolic adaptations that reduce photorespiration. The two most important of these adaptations are C4 and CAM plants. C4 plants have two kinds of photosynthesis cells, bundle sheath cells and mesophyll cells. The bundle sheath cells are grouped around the leaf's veins, and the mesophyll cells are dispersed elsewhere around the leaf. CO2 is added to phosphoenolipyruvate to form the 4-carbon compound oxaloacetate. This reaction is catalyzed by PEP carboxylase, which does not combine with oxygen and does not participate in photorespiration. At this point, carbon is fixed. Whoa!
Jeez!
The mesophyll cells export the oxaloacetate to the bundle sheath cells, which break down the oxaloacetate, releasing CO2. The C4 pathway then acts as a CO2 pump.
Oh god. Oh jeez.
The CO2 in the bundle sheath cells is then converted into carbohydrates through the normal Kelvin cycle. In C4 plants, the mesophyll cells pump CO2 into the bundle sheath cells, keeping the CO2 concentration high enough so that Rubusco will be likely to bind the CO2 rather than the O2. This reduces phosphorespiration. In C4 plants, the two stages of photosynthesis are separated structurally, providing a process that minimizes photorespiration and enhances sugar production. Now, for CAM photosynthesis, which is another adaptation to the hot, dry climates like me, the desert, these plants keep their stomata closed during the day to prevent excessive water loss. Of course, this also prevents gas exchange. At night, the stomata open, and CO2 is fixed in organic acids and stored in vacuoles. The morning, when the stomata close, the plant cell release the stored CO2 in the form of acids and proceed with photosynthesis. In CAM plants, these two stages of photosynthesis are separated temporarily. In both C4 and CAM photosynthesis, CO2 is first transformed into an organic intermediate before it enters the Kelvin cycle. All of the processes, C3, C4, and CAM photosynthesis, use the Kelvin cycle. They just have different methods for getting there. What?